We have one other student here. Oh, there's a few coming out now. All right. All right. Sorry about that. I was in the wrong place. I was in the lecture for some reason. Usually when I end the lecture button disappears, but apparently I had gone into the lecture and was waiting for students to be there. Thank you for the student who went to the lecture and then told me I was in the lecture because I didn't even know that. You're not marked down. All right, let's get going. Let me share my screen here. Where are we? Sorry, I'm lost. There it is. All right. You should be seeing my screen, although there's nothing much here. Let me go home here. So what I thought we would do is work backwards through the syllabus, if I still have it up here. So I want to cover a little bit of chapter 17, although we just covered it, and then chapter 13 and chapter 14, and maybe a little bit of lab 9, because this will be major emphasis over this material on the final. The final is comprehensive, but the most questions that will be answered would be chapter 8, and there's like 3 questions from chapter 8, 3 or 4. And then for like chapter 2, I think there'd be only 1 question. And then chapter 1, we'll spend a lot of time on chapter 1 as well, because chapter 1 is a chapter that introduces a topic, and then we later came and discussed a lot of those topics. So chapter 1 does introduce a lot of topics. So we'll spend more time on chapter 1, maybe a little bit more on chapter 8, but a lot of time on the last chapters we just covered. Any question about any of that? So before I begin, let's begin with lab module 9. Oh, wrong one. Come on. So identification of gram-negative bacilli. You should know how to read the indole test, the methyl-red test, the Voges-Perskure test, and an easy way for remembering these is all 3 of these, when they're positive, they will be red. You should also know how to read the citrate test, and if it's positive, it will be blue. For the indole, methyl-red, and the Voges-Perskure test, if it's negative, it'll be a yellowish color. For the citrate test, if it's negative, it'll be a green color. You should know how to do carbohydrate fermentation, and that one we've done a couple of times. So this isn't the only lab we've discussed that. It will be yellow if it is positive, and it could be yellow with a gas bubble if it's acid gas. Acid is just yellow, which both of those are positive. So I shouldn't call it positive. I should call it either acid or acid gas. Negative would be pink, and I can't show you that there, but let me see if I got it down here. Negative, positive, positive with an air bubble, meaning acid and acid gas. All right, you should know the motility. It's pretty easy. Also, just you're looking for is the growth only in the stab line. That's non-modal. If the growth is everywhere, as shown here, it'll be cloudy, not clear like that on the sides. That's very modal. If it's slightly modal, it will be flaring out from the stab line. And the flare will just be, I don't know, flares, whatever you want to call those. Any question about motility? You should know hydrogen sulfide production test, whatever test you're running. If it shows hydrogen sulfide was produced, it'll turn black. So this looks like it's a Sims tube, and it's black because it's positive. Not black is negative for hydrogen sulfide production. Now, the next one is a little more difficult, and this one I will admit is difficult. And I wish I had an easy way to describe it. Explaining the triple sugar iron test, TSI. And you're looking at the growth both anaerobically when you inoculate the tube, and then aerobically on when you smear the slant. So this is aerobic growth, this is anaerobic growth. Let's just go to the results. If the tube is all pink or orange, that's a negative result. And you should say that negative on the slant over negative on the butt. And there they're not using negative, they're using K for negative. K means alkaline reaction on the slant over the butt. So for the TSI, you got to go both slant and butt. Sugars are not produced. Why it's negative and negative. No hydrogen sulfide being produced. That'll be black. Here on tube, we have hydrogen sulfide being produced. Because it's black here, you kind of have to know this. It only produces hydrogen sulfide when the sugar is fermented. So at least in the butt, the color would have been yellow if we could see the yellow through the black, or if you looked at the tube earlier, you would have seen like yellow around the edge and black in the center. Any question about that? Now, in the case of glucose only being fermented, the slant will turn red or pink because the glucose is at low numbers in the tube. And aerobically, the glucose will be metabolized quickly. The growth will happen quickly. Anaerobically, the sugar will be metabolized slower and the growth will be slower. So if it's yellow on the bottom and red or pink on the slant, only the sugar glucose is metabolized. And if we looked earlier at the slant, that would have been yellow, but all the sugar has been used up. Rather than dying, the bacteria use protein. And when they use protein, they make ammonia. And that is basic, ammonia is basic, and that changes the color of the slant from yellow to red. So this is K or negative on there, and then acid down here, and we know that that means only the sugar glucose is fermented. It was fermented here, but then it's all used up. Any question about that? No hydrogen sulfide being produced in any other tube except for two. This tube, if it's yellow on both the slant and the butt, then we know that both glucose and at least one other sugar is fermented because the slant is yellow. And we don't know which sugar it is. It could be both glucose and lactose or sucrose. or it could be sucrose only and glucose. Glucose is always fermented if one of the other sugars is fermented, because glucose is the base sugar. These other two sugars are actually converted into glucose and then sent through aerobic respiration in the normal way. This tube also has gas production. You can see a bubble or the cracking of the media, which means production of gas. That's how you read the TSI tube. And yes, it's complex. Sorry about that. The SIMS tube, you stab inoculate it. If it's yellow on the top, then there's no indole being produced. If it's pink on the top, then the Kovacs reagent is added. And if it turns pink, then indole is produced. SIMS stands for sulfide production, hydrogen sulfide. And if it's black, hydrogen sulfide is produced. I for indole being produced. And then M for motility. You stab inoculated. If you only have growth in the stab, then it's non-modal. If it flares from the stab, as this one is doing, then it is modal. Any question about that? McConkey's and HE plates look at growth, and if it grows, it's likely to be a gram-negative cell, gram-negative bacteria. With McConkey's, you can look at lactose fermentation, and if the sugar lactose is fermented, it'll be pink, both the colony growth and the media around it. And if it's not fermenting the lactose, the bacteria will be its normal color, whatever color it is. In this case, it's white or off-white. For hectone enteric agar or HE plates, if it ferments the sugar lactose, it'll be salmon colored. That's a yellow-orange color. If it does not ferment the sugar lactose, the bacteria will be the normal color, which here is white or off-white. If it grows on HE, I forgot to mention that, then it's likely to be a gram-negative cell. So these are all gram-negatives. And then if it's black on hectone enteric agar, regardless of whether it ferments or does not ferment, if it's black, that means it's forming hydrogen sulfide gas. Any questions about that? All right. From this lab, you should know how to do case studies. I may have a case study. I would only have a very simple one in a multiple test exam because you don't have a lot of time to work on the problem. But I could give you a case study in a what do you call it? A non-timed exam. I think there are a couple of questions which are non-timed, in which case there could be a case study in the non-timed exam, which would follow something similar to the case studies in Lab 9. Any questions about Lab 9? All right, if not, let's go to Chapter 13. The main thing is to know the terms, obviously. Be able to discuss the factors that influence microbial death rates. And here it is here. The more microbes, that influences the microbial death rate, and you need longer exposure, time of exposure of the agent. So these two, time of exposure and number of microbes, go together. The more microbes you have, the longer the time of exposure you need to kill the microbes. Environmental influences like the presence of organic matter can influence the microbial death. And then we never got to microbial characteristics, so I'm not going to be able to test you on that. So you don't need to know about that one. Be able to discuss the three main ways in which antimicrobial agents kill or inhibit cells. They can alter the plasma membrane, like put a hole in it. They can denature proteins and enzymes that'll kill cells. And then they can mutate or damage DNA. Those are the three main ways that antimicrobial agents kill cells. You should understand some of the types of disinfectants and how they work. We did discuss all of the physical agents for killing microbes. Like UV light forms thiamine dimers, ionizing radiation forms ions. Five, you don't need to know because we never got to it. The relative resistance of the major microbial groups to antimicrobial agents. Any questions about chapter 7? Just looking to see if there's anything I want to discuss. You should know about pasteurization. There are two methods to pasteurize milk or other agents. 63 degrees Celsius for 30 minutes, 72 degrees Celsius for 15 seconds. And remember, ultra high temperature milk is not pasteurized milk. That's actually sterilized milk. All right, any questions about chapter 7? If not, that's it. For chapter 7, let's look at chapter 13. Chapter 13 is viruses, viroids, and prions. Let me just say you should know what viroids are and what prions are. You should know how prions reproduce, and that is they're a protein. And how they reproduce is they go up to a normal cellular protein and then cause that protein to transform into the prion protein. And you have to realize that prion protein has the same amino acid sequence as the normal PRP superscript C protein in the nerve cell. So I guess you should say prions are really a disease of nerve cells because they're the ones who have the PRP superscript C protein. And then you should know about viruses. With viruses, you should know what their shape is. They obviously have a genome, which can be DNA or RNA. They can have a capsid around it, and they all have both of these two. If the virus only has a capsid, then we call it a naked virus. But some viruses have an envelope around it, like HIV, and you should know how the cells obtain that lipid bilayer. It's actually a part of our cell's membrane. You should know bacteriophages are viruses which infect bacteria cells. They have two life cycles, the lytic life cycle and the lysogenic life cycle, and you should know both life cycles. You should know what a prophage is and a provirus. You should know the different types of infections in animal cells, like a latent infection, a persistent viral infection, a budding life cycle, and then, of course, animal viruses have the lytic life cycle as well. All right, understand about characteristics and classification of viruses. The viral multiplication life cycle I've already discussed, and we already talked about prions and virions. You should know that viruses outside of their host cell are metabolically essentially not metabolic. They are inactive. And they're only metabolically active. They only grow and reproduce when they have infected their host cell. Their host cell is very specific, and most viruses only infect a few cells of one or two species. Like HIV only infects a few cells, the helper T cells, I think macrophages, I think brain cells, maybe nerve cells, but do not infect other human cells. And they do infect one other species, one of the chimpanzees, but it doesn't cause AIDS in the chimp, only in humans. Viruses have no ATP generating mechanism. They rely on their host cell for supplying a lot of things for them, including enzymes, nucleic acid, amino acids, and ATP. Viruses are very specific in their host range, and that they have attachment molecules called ligands or adhesions that bind to a host cell molecule, and that host cell molecule is called the receptor. And that's why most viruses are very specific and can only bind to certain cells in one or two species. Now, there are some viruses that do infect many different species. And obviously their host range is very broad. Eastern equine encephalitis, for example, tends to be a virus which can infect most mammals, obviously humans. And one of the equine encephalitises, if not all of them, I don't remember, but one of them can infect birds. Bird flu is another virus. It can infect mammals and people, but it mainly infects birds, and it can infect many different species of birds, if not all species of birds, and then many species of mammals. So not all species are very specific, but many of them are. You know, the relative size of the viruses. We already talked about that. Talked about that and that. You know, the different morphologies of viruses. They take three basic shapes. A capsid is either a helix, a polyhedral, or then a complex virus, which can take on any shape. You should know about viral taxonomy. We aren't able to group viruses into much above a family. You can group them into, let's see, King Peter's son came over from, you can group them into orders, but you can't group them much higher than that. We mainly group viruses into families, and the virus family will be Indinviridae, like herpes viridae. And then there's the virus genus. The herpes virus is the genus name. So the genus name ends in virus. And then the viral species, we use the common English name, herpes, simplex type 1, common viral species name. And then the subspecies is designated by the number. And in viruses, the subspecies is very important. Obviously, herpes simplex type 1 and herpes simplex type 2 is the same viral species, just a different subspecies, and it causes generally different diseases. And then a better example is with the polio virus. Polio virus type 1 causes the disease polio. Polio virus type 2 is the vaccine that we use to prevent getting the disease polio, meaning we vaccinate people with the polio virus type 2 because it doesn't give the disease and it protects you from getting the disease polio. Any question about that? All right, I've already talked about the lytic life cycle and the lysogenic life cycle. Let's briefly go over the the lysogenic life cycle. And that is because when a lysogenic virus first infects a cell, it has to immediately decide which life cycle to enter. It may enter the lytic life cycle, where the virus will replicate, lyse the cell, and then that will release the newly replicated virions. On the other hand, if the virus enters the lysogenic life cycle, the viral genome will incorporate into the host cell's chromosome. And then the two will live together in symbiosis for as long as the virus stays in the lysogenic life cycle. When the viral genome integrates into the host DNA, we call this a prophage, the virus inside the host DNA. Whenever the virus decides to leave the lysogenic life cycle, the prophage will be excised from the chromosome, and then the virus will then enter the lytic life cycle. Any question about that? All right, there are some consequences for lysogeny. The most important is a bacteria that has a lysogenic cell gets, lysogenic virus in it, gets new DNA, which will be the DNA from the virus, and that can express new genes, causing the phenotype of the cell to change. So this cell, before being infected with a lysogenic virus, could not produce the diphtherium toxin. However, Corynebacterium diphtherium, once it's infected with a lysogenic phage, can produce the Corynebacterium diphtherium toxin. Any questions about that? All right, animal viruses generally multiply the same as bacteria viruses. There are some differences. The main difference is simply that animal viruses may replicate in the cytoplasm, which is where bacteria viruses replicate, but animal viruses may also replicate in the nucleus. As a rule of thumb, a DNA animal virus will replicate in the nucleus. I don't know of any exceptions. As a rule of thumb, an RNA animal virus will replicate in the cytoplasm. There is one exception, the family Retroviridae. It is an RNA virus, and it replicates in the nucleus. The reason why? The family Retroviridae has an DNA intermediate. Any questions about that? All right, so we should know that animal viruses can replicate by the budding life cycle, and that is how an enveloped animal virus obtains its envelope. It's a part of the membrane of its host cell. It could be the cell membrane, the nuclear membrane, or the endoplasmic reticulum, wherever the virus is budding out of, meaning it could bud out of the endoplasmic reticulum. Certain species do, and most of them do bud out of the cell membrane. For example, I think HIV buds out of the cell membrane. You should know that if an animal DNA virus integrates into the host DNA, host cell's DNA, we call that a provirus. And once that integrates, it becomes a permanent genetic feature of that cell for as long as the cell is alive. For the family of retroviruses, you should know that they're special because they have the enzyme reverse transcriptase, which can make single-strand DNA out of RNA and then can make double-strand DNA out of single-strand DNA. Okay, you should know about COVID-19. It's a coronavirus. You should know that viruses can cause cancer by one of two ways: the formation of a provirus or by bringing in an oncogene to the cell. All right, any questions about chapter 13? Let's move on to chapter 14. In chapter 14, I already know you should know the terms of this chapter. If you know the terms of this chapter, you'll learn 50% of the material in this chapter. You should also understand the relationship between the normal microbiota and its host. Understand the stages that disease goes through in the development of disease. And then, most importantly, understand about nosocomial infections. It's most important because most of you are going to work in the healthcare industry, so you should know what nosocomial infections are and how to help prevent spreading them, how to help prevent your patients from getting a nosocomial infection. Any question about any of that? All right, you should know about the normal microbiota and where it's found, essentially on the entire outside of the body, the skin, the mucous membranes open to the outside of the body, the gastrointestinal tract, all of it. The respiratory tract, only the upper portion usually has normal microbiota. The lungs do not usually have microbes in them. And then it used to be said that the bladder did not normally have microbes in it. But as I've told you, a recent finding is they found, at least in females, normal microbiota in the urine in the bladder, at least of females. So the lower genital urinary system does have normal microbiota. The upper genital urinary system does not normally have normal microbiota. And don't ask me where the upper is because I suppose it depends if you're a male or a female, but the bladder is one region where you can have a normal microbiota, at least of females. The kidneys would be definitely where you do not normally have normal microbiota. The gastrointestinal tract, I don't know if I mentioned that, totally coated with normal microbes. The normal microbiota are organisms that generally colonize the host at an early age, like maybe birth, and continue to live with the host for essentially the host's entire lifetime. That's different from a transient microbiota, which may be present for days, weeks, or months. And I gave an example where for me, Staphylococcus aureus was a transient microbe. I had it for one time for, I'm not sure, maybe a couple of weeks after I hugged well, actually my aunt hugged me and she had Staphylococcus aureus skin infection. Any question about that? Fortunately, it was only transient. My normal microbiota threw it off eventually. I guess you should know there's more normal microbiota cells than there are human cells in the human body. And then you should know that the bladder as well as the female human breast has microbes, normal microbes in it. And so when the female makes mother's milk, before it ever leaves the mother, the milk has already got normal microbes in it. And then the placenta harbors a unique microbiota found only in the placenta. You should know about microbial antagonism. The normal microbiota can engage in microbial antagonism with a human pathogen, preventing the human pathogen from getting established. They can do this by occupying the niche the pathogen's trying to occupy, by competing for resources, by making the environment less favorable to the pathogen. And this is usually by producing acids in both the mouth and the urethra and the skin. All of these, the normal microbiota make it more acidic. And then normal microbes can also produce a bacteriocin. This is a protein that acts similar to an antibiotic. The only difference is it's more narrow in its spectrum of action. For the bacteriocin being produced by E. coli, it will only inhibit the growth of Salmonella and Shigella. It would not inhibit the growth of other microbes in the gut, such as, oh, I almost had it, Enterococcus faecalis. It would not inhibit that one. Antibiotics, if you were to take that, it would inhibit most of the microbes in the gut, because the most narrow antibiotic you could have would be one working on gram negatives. And that would affect most of your microbes in the gut. All right, so your normal microbiota forms a symbiotic relationship with us, and it's either commensalistic or a mutualistic symbiotic relationship. Mutualism is where both organisms benefit. For example, E. coli gets a home, we feed it food. E. coli helps us digest the food that we eat. So E. coli is giving us, is forming a mutualistic symbiotic relationship with us. Commensalism is where one organism benefits, like the nematode in our eye is given a home, but the nematode in our eye does not cause us any harm. So it's a commensalistic relationship. On the other hand, a pathogen forms a symbiotic relationship with us, but it is a parasitic relationship where one organism benefits, the pathogen gets a home, and then it gets nutrients from us, and it damages us. And that's why it's a parasitic relationship. You should know about opportunistic microorganisms. Some normal microbiota can cause infection under certain circumstances, in which case we call them an opportunistic pathogen. Like E. coli is normally found in the gut, but if it is growing in the inner ear, that would be an opportunistic pathogen, an opportunistic microorganism. Any question about any of that? All right, you should know about Koch's postulates. We've talked about it at least 3 times in the term. The steps you use to show that a given microbe causes a given disease. First, you isolate the microbe from the sick individual, isolate it out, grow it up, and then purify it, obviously, and inject a pure culture into an organism or another patient. If that patient becomes sick, that's an indication that this microbe causes the same disease. But then you also have to show that you have that microbe in the sick individual. And if all of these steps are met, then you can say that this microbe causes this disease. And Robert Koch used this to show that Bacillus anthracis causes the disease anthrax. the first bacteria shown to cause disease. Any question about any of that? You should know there's exception to Koch's postulates. And what we do is we use vast epidemiological studies when we cannot use Koch's postulates. You should know these terms. You should know what a latent disease is, herd immunity. Patterns of disease. In order for disease to spread in the human population, the disease has to come from a reservoir. The pathogen must be transmitted to a susceptible host. The pathogen must invade the host and multiply in the host. And furthermore, to cause disease, the pathogen has to injure the host. If there's no injury, there really is no disease. The illness will then end when A, the host immune system destroys the pathogen, B, the pathogen kills the host, or C, I guess the pathogen could establish a plateau where the pathogen is persisting for a long period of time. An example would be cytomegalovirus. You should know about the predisposing factors, making it more susceptible to getting a disease. You should know about the stages of disease development. Before an individual becomes infected, they have to get infected by the organism. And before any signs or symptoms appear, we call that the incubation period. Following the incubation period is usually the prodromal period, where the signs and symptoms are mild. That's followed by the most severe signs and symptoms of the period of illness, followed by the period of decline. For example, a patient who has a fever, when the fever breaks, that's when they've entered the period of decline. The number of microbes then decrease, and eventually they fall below a certain threshold where you have the microbes, but they no longer cause disease in you, in which case we call it the convalescence period, where yes, you still have the microbes, but they are no longer causing any signs or symptoms of disease. Any question about any of that? Now, not every disease has every period of illness. For example, the flu may not have the prodromal period. HIV might not have the period of decline or the period of convalescence. Any disease-causing organism, if it kills the patient, there is no period of decline or period of convalescence. All right, you should know about the spread of infection. And that is a pathogen has to come from a reservoir of infection. There are three types of reservoirs: human reservoirs, animal reservoirs, and then non-living reservoirs. You should know about the transmission of disease. It can be contact transmission, which could be direct contact transmission, indirect contact transmission, or droplet transmission. Direct is when one person directly touches another. It could be kissing, handshake, high-fiving. Indirect, an object or fomite receives the pathogen from, let's say, in this case, the catcher. The baseball is the fomite, and then the catcher touches the baseball, picking up the microbes. And usually the catcher or other individual has to inoculate the microbes in a mucous membrane in the face. So the baseball does not give it directly, it gives it indirectly, and then the patient has to further spread it to their mucous membrane. On the hands, would not likely cause disease. There are some exceptions, like maybe Staphylococcus aureus could cause a skin disease on your hand, but most diseases, you have to inoculate the mucous membrane. I'm not going to talk about droplet transmission. That's the third contact transmission. And the reason I'm not going to talk about it is airborne transmission is not direct transmission, and yet the same sneeze can be either droplet transmission or airborne transmission, which is a form of vehicle transmission. There are three forms of vehicle transmission: waterborne, foodborne, and airborne transmission. And then there's vector transmission. That is where an animal brings the disease to the individual. The biting arthropods are the most important animal vectors bringing disease. A good example would be the mosquito bites the human, giving the malaria, the Plasmodium species, to the patient, and then the patient then develops malaria. All right, know about nosocomial infections. That about 1 in 10 hospital patients will get them. They happen because microorganisms are present in the hospital or hospital-like setting, like a nursing home or something like that. The patient in the hospital tends to be a compromised host, one who's more likely to get disease just because he's compromised. And that could be broken skin, broken mucous membranes, burns, injections, ventilators, catheters, anesthesia. All of these weaken the host and make it more likely to get an infection. And then there's a chain of transmission in the hospital or hospital-like area. There can be direct contact transmission, patient to patient. There can be indirect transmission via fomite, such as a hospital patient turning off the light switch, and then another patient comes in and turns on the light switch. That could spread disease. And then there can be vehicle or airborne transmission. Obviously, COVID-19 is spread mostly by vehicle or airborne transmission. You should learn how to control nosocomial infections. All right, any question about chapter 13? If not, let's quickly go on to chapter 14. I already did chapter 14. Let's quickly do chapter 8, and I'll quickly do chapter 1, and then we'll break. In chapter 8, you should know the terms. You should be able to describe DNA replication, RNA transcription, and protein translation. You should understand about the different types of mutation. Oh, and you should understand about the regulation of bacterial gene expression using an operon. Just quickly going through here to see if there's anything I want to talk about. Yeah, you should explain how an operon, you don't need to do it totally, but I might ask a question on how an operon is blocked, like a lac operon, and then how it becomes activated by allolactose, which is around whenever the sugar lactose is around. So you should know about base substitution, both a silent mutation, a missense mutation, a nonsense mutation. You should know about frame shift mutation, and you should know about macromutation, where an entire region of the chromosome is added or deleted. And let me remind you that macromutation is not discussed in the book. And about half of you got it wrong on the in-class activity for chapter 8 too. I'm saying half because in my other class, there were a lot of people who answered this question because there's a lot of people in that other class. All right, any questions about chapter 8? If not, let's quickly go through chapter 1. Sorry, I'm going over here. All right, you should know the terms. You should know about the brief history of microbiology. And in the history of microbiology, you should be able to tell me what was a major invention, or tell me about the major inventions, and then who discovered it. In the history of microbiology, you do not need to know any of the dates except for the first one, which was when the first fossil record were discovered on the planet Earth, how old they are. So how old the oldest fossils are. Where is that? I'm not finding where that is now. Oh, here it is. It's right before this. There we go. So greater than 3.5 years ago, we had the first evidence of life, the first fossils. Any question about the history of microbiology? All right, you should discuss the importance of microbes. Like the importance of microbes in the nitrogen cycle is most of the steps of the nitrogen cycle are happening because a microbe is doing that one step. So a microbe is fixing bacteria from the air, bringing it into, I guess, our ecosystem. And most of that, when after it's brought into like a microbe, when the microbe dies, and then most of it will be changed into ammonia. Ammonia can be changed into nitrite, nitrite into nitrate. All of these are happening by a microbe. And then the nitrate is what green plants prefer to use. And then they'll use nitrate for their organic nitrogen source. And then eventually they'll die, releasing it into ammonia, starting the cycle again. And then nitrate can be converted back into nitrogen gas in the atmosphere. You should know that very few microbes are pathogenic, only about less than 2%. Microbes play many beneficial roles in the ecosystem into humans. You should know that we do classify all living things into one of three domains. The domain is defined. You can find the domain just by sequencing the DNA of a species. And if it belongs to that domain, then that species will be closer to every member in that domain than to any organism outside of that domain. And there are three domains: the bacteria, the archaea, and then the eukarya. The archaea and the bacteria have the phenotype prokaryotes, And that's because they're simpler than eukaryotes. Eu means true, karyote, we translate that for nucleus. So eukaryote means true nucleus. You should know that there are two cell types. That's the term your book uses. And that is a prokaryote is one cell type. The other cell type is eukaryote. You should know about Louis Pasteur's famous swan neck experiment, where he took a glass beaker with a very long neck, poured the media in, heated the neck, bending it in S-type shape, and then sterilizing the media, and then letting it cool. And the microbes from the air do not fall into the Swan Lake nest. All right, any questions about chapter 1? If not, I'm going to end it there. I'm sorry we didn't get to discuss the other chapters. There will be a few questions on all the other chapters, the exception being chapter 1, where I think there's only one question. Not chapter 1, chapter 2. I think there's only one question in chapter 2. All the others will be a few questions, but not more than 3. Any questions before we go? All right, good luck on your phone.
Sorry, just 1 second.
Yeah.
I have a question. I was wondering, if the grades will be in for the infectious disease, project and the, lab report before we take the final.
Probably not. I'm still working on the labs. I'm still working on lab 01, grading it for the second time. I hope to finish the labs by this weekend and then work on the bigger 2 projects, but I don't know if I'll finish them. Normally, with only 20 students, it takes me a day to finish each of those, a solid day, 24 hours working. So let's say 2 days worth. For 20 students, I've got 60 students this term. So my guess is it'll take at least 6 days to get those big assignments finished. So I doubt I'll have them graded by the time of the final. I will try. Like I said, my goal is to get the labs graded by this weekend and then begin working on the bigger assignments, but I'm not sure when I will get them graded. Sorry.
Okay, all right. I just thought it would help to gauge like the
How you're
final.
doing. Yeah, I
Yeah,
understand.
because those are big. Those are a lot of points.
Yes, they are.
So, all right. Thank you.
All right. Bye.